Welcome to BioTek's quantitative assay apps. These applications can be used to determine protein or DNA concentrations within laboratory samples in conjunction with BioTek's absorbance and fluorescence plate readers. Once installed, you can launch the app by clicking on the icon from your Windows desktop. The application will open with a new workspace. To import settings from a previous session or open an existing file, you may select these options from the drop-down menu in the top left. For demonstration purposes, I'm going to walk through the setup and analysis of a BCA protein quantification assay. The first step is to choose the desired assay from among the installed applications from the drop-down on the left sidebar and selecting BCA. In order to define my plate layout, I will start by clicking and dragging over the wells that contain my blanks. Next, I will click and drag over all of the wells that contain the known protein standards. This will open up a dialog box for entering the concentrations of the standards. Notice that the number of standards and replicates are already inferred from the selected area. Within this dialog box, I have three options for entering the standard concentrations. I can enter the concentrations in manually. I can use the default concentration values for the particular assay. Or I can enter an initial concentration and a dilution factor, either dividing or decreasing by a certain value. Once I click Assign, the selected area will fill with the designated sample concentrations. Finally, to complete my plate layout, I will click and drag over the area containing my samples. Here I can indicate the number of replicates per sample and whether replicates proceed along rows or down columns. If sample dilutions are present, these can be included here as well, again indicating whether sequential dilutions proceed along rows or down columns. Lastly, I can indicate whether my samples are stacked along columns or adjacent to each other along rows. By clicking Assign, the selected area will fill according to the pattern indicated. Here I see sample 1 in the 4 dilutions of sample 1. And sample 2 adjacent to it along the same row in its dilutions down the column. At this point, I'm ready to begin my read. I will place the plate on the instrument and press Start Reading to initiate the absorbance measurement. Immediately following the completion of the read, I must review and approve the standard curve. I can choose between a linear or quadratic fit. The R-squared value can be used to assess the quality of fit between the two methods. If necessary, individual standard replicates can be masked. If data points are masked, the current fit with the masked points excluded appears in blue, while the original fit, including all points, appears in gray. Once satisfied with the fit, approving the standard curve by clicking Approve Curve will allow for the estimated concentrations of the samples to be calculated. If I wish to save a copy of the graph, I can specify a folder for the PNG file to be exported to. Reviewing the Data tab shows the results in a plate orientation. The values within each well indicate the sample, the optical density, and the estimated replicate concentration. Reviewing the Statistics tab, the estimated concentrations are shown, accounting for sample dilutions if included, as well as summary statistics for each group of replicates. In this data, I can see that the 1 to 8 dilution of the samples deviates from that of the other sample dilutions. Dilution of these samples to this extent has brought them to the lower end of the assay detection range. Therefore, I can mask these replicates from the dataset. Returning to the Data tab and clicking Mask Samples, I can drag over those replicates which I wish to exclude. Now on the Statistics table, I can see that the 1 to 8 dilutions have been masked. Finally, on the Normalization tab, the mean concentration for all samples is calculated across all replicates and dilutions if present. By default, the normalization factor calculates the proportion of all samples to the least concentrated sample, thereby indicating the full dilution required to match the concentration of all samples to the least concentrated sample. However, if I wish to normalize my samples to a target protein concentration for a specific application, this concentration can be specified and the normalization factor will update accordingly. Here I'll use 300 micrograms per mil as the target concentration. I can now see that sample 4 is 3.34 times as concentrated as my desired concentration. Or said another way, sample 4 will have to be diluted by a factor of 3.34 in order to achieve my target concentration. Similarly, if there is a specific volume I would like my normalized samples to be in, this target volume can be included as well. For instance, if I need my samples to be in a final volume of 25 microliters, I will need 7.48 microliters of sample 4 diluted to 25 microliters to achieve my target concentration. Using these numbers, I can immediately proceed with diluting my samples. If I wish to save the values from the Statistics and Normalization tabs, I can click on the Export Data button and specify the location for the TXT files to be placed. If I would like to save the Gen 5 experiment file, including the plate layout, plot, and results to review again in the future, I can do so by going to the drop-down menu and choosing Save As. If you have any questions or require support, please contact BioTek's Technical Assistance Center.